Hello everyone, I'm Siyuan Shen from Central South University, and today I'm excited to share our work titled Precise Prediction of Hotspot Residues in Protein-RNA Complexes using Graph-Partition Networks and Pre-trained Language Models. Let's begin with a bit of background. Now, protein-RNA complexes are involved in essential biological processes like splicing, translation, and gene regulation. However, only a small number of residues, so-called hotspots, make major contributions to the binding energy. These hotspots, shown in the red here, are especially valuable as drug targets due to their functional importance. But the challenge is experimental techniques like aniline scanning are slow and expensive, and existing computational tools often rely on 3D structures which aren't always available. So our motivation was clear. Can we develop a method that predicts hotspot residues from sequence alone without needing high-resolution structures? That's exactly what we set out to do with deep hot residues. Now, here's a quick walkthrough of our model. We start with the protein sequence and predict its 3D structures using ESM fold. From that, we build a contact map by connecting residues within 10 angstroms. Meanwhile, we extract 4 types of residual features: DSSP, PSSM, HMM, and ESM2 embeddings. They are combined and pass through a squeeze and extraction module to rewrite important channel. Then we use the graph-partition network where residues as are nodes and each reflect special proximity. Finally, we get per residual scores highlighting the likely hotspot residues. Let's take a look at the dataset. We collect mutation-based binding data from 147 protein-RNA complexes, covering 835 residues in total. Data come from literature, prior studies, and the NABI database. And by the way, the NABI database was created by our lab specifically to collect and organize experimental hotspot annotation. To reduce redundancy, we filtered out similar sequence and ensure balanced hotspots and non-hotspot label. And after filtering, we ended up with 99 complexes and 283 hotspots and 426 non-hotspots. One big challenge we faced was class imbalance. Hotspot residues are much fewer. So inspired by Shah et al., we used a label transfer strategy. Here is how it works. In step 1, we select protein pairs with TM score greater than 0.5 and the sequence identity greater than 40%, ensure both structural and sequential similarity. And step 2, we map hotspot labels from protein A to align residues in protein B. This approach help us expand the hotspot class without introducing noise. Next, let's take a look at the result. We evaluate our method using metrics like accuracy, precision, recall, F1, and AUC. Under 5-fold cross-validation, the model performs very well. The AUC reaches 0.956. On an independent test set, the compared deep hot residue with 4 other models: XGB, PRH, Prep Hot, Hot Spring, and Spray Hot. Our model outperforms of them, especially in the AUC and specificity. To further evaluate our model, we compared it with 7 baselines, including traditional models like SVM, Random Forest, XGB Boost, and deep models like DNN, GCN, GraphFormers, and GraphGPS. According the board, our model consistently outperformed our baseline. It achieved the best precision F1 and AUC. And we also performed a feature abortion study to see how each input type contributions. Removing ESM2 embeddings caused a huge drop in performance. This clearly shows how crucial pre-trained language model features are. Removing DSSP, HMM, and PSM features also hurts the performance, but not as dramatically. So overall, ESM2 is the backbone of our feature representation. Next, we want to see if the model's prediction makes structural sense. Previous studies have shown that hotspot residues tend to be buried, high hydrophilic, and partial solvent exposed, so they are often surrounded by contacting residues, forming an O-ring structure. To validate our model capture this pattern, we use PyMo to visualize the 3D spatial environment of high-confidence predictions. As shown in the figure, the prediction hotspots are spatially clustered and often lie adjacent to non-functional residues, consistently with experimental verified hotspots. Now, a quick note at the model scale. We test 5 versions of ESM2 from 8 million to 3 billion parameters. And as you might expect, large models performed better across all metrics. The 3 billion version was the best, suggesting that bigger really better when it comes to capture complex protein features. Finally, we did a case study on MS2 called Protein-RNA complexes. And experimental scanning found 4 hotspots and 6 non-hotspots. Our model got all 4 hotspots right and 5 out of 6 non-hotspots. Other models struggled. Prep hot got 3, hot spring got 2, and the spray hot got only 1. This shows our model performs well even on real biological data. So this is conclusion. To summarize, our work focused on identifying hotspot residues in protein-RNA complexes. We developed a method called deep hot residue, a sequence-based deep learning model that, using ESM4 to predict protein structure and the contact map, extracts multi-source features like PSSM, HMM, DSSP, and language model embeddings, enhance them with a SE module and use graph fetish network to predict hotspots. In experiment, our model achieves a SOTA performance in five-fold validation. So thank you for your attention.
Thank you to the speaker. Are there any questions? Okay, thank you for the talk. I'm just curious about a distance. You show that you are selecting 10 angstrom in the very first slide that limited. And then when it comes to feature extraction, is there any reasons behind why you set 10 angstrom? Is because a protein-RNA context?
Yeah, we test from 2, 4, 6, 10, and 12. These numbers has used in previous studies, so we tested them and we found that 10 angstroms is the best performance in our model. Thank you.
Yeah, it seems like AlphaFold3 can also do something similar. Did you compare with it?
Yep, but when we do this work, the AlphaFold3 is not. When we do this work, the AlphaFold3 is not open source, so we didn't use it.
Okay, but did you compare somehow later and to see? Because if you want to use it, you have to show. Do you have comparison result now? Maybe at the time you submit it's not, but maybe.
Yep. We can consider do it later.
Okay.
Thank you.
Thank you for the talk. I would like to ask if you could elaborate a little bit on the build of the adjacency graph and on how many layers in the graph attention transformer will you use? Like, I imagine that you need to choose how far away would be the node, like how many neighbors for each node to build the adjacency graph. So could you elaborate on that a little bit?
I can't stand. Maybe you can ask again.
Okay, yeah, sure. Could you please elaborate on the building of the adjacency graph that you use as input to the GATT block?
Sorry, I cannot understand this question. Maybe you can send the question to my mail.
Yeah, sure. Yeah, sure.
Okay, sorry.
Okay, thank you.
Any other questions? Well, in that case, we can thank the speaker again, and we can thank all the speakers of this session.
Okay, thank you.